At Broughton, we understand the regulator's needs for clinical and behavioural data as part of a robust scientific evidence package to support the regulatory submissions of next-generation nicotine products, or NGPs. Whilst preclinical data demonstrates the risk reduction potential of an NGP, the clinical and behavioural studies begin to answer questions around the likely use scenarios of the products, especially the likely attractiveness and potential use patterns of the product amongst the intended audience, current smokers, and amongst non-intended audiences. In essence, these data begin to provide information around the off-ramping and the on-ramping potential of these products, where the desired outcome is to provide evidence that the new product will maximize smokers off-ramping from cigarettes and minimize on-ramping into nicotine for current non-nicotine users. Clinical studies for NGP products invariably always incorporate a pharmacokinetic and pharmacodynamic approach. These studies are important to determine if the product is sufficiently satisfying for current adult smokers to switch to, and to determine if the delivery of nicotine from the NGP is similar to a cigarette. When we talk about nicotine delivery, this is really to determine the maximum concentration of nicotine in the blood, Cmax, and the time taken to achieve this maximum concentration, Tmax. In order to ensure that the NGP is not delivering nicotine at a level which makes it more addictive than a cigarette, the Cmax of the NGP should not exceed that of a cigarette, and the Tmax should not be shorter than that for a cigarette. In the ideal scenario, the Cmax and Tmax would be the same as cigarettes to maximize the success of off-ramping for current adult smokers. Some manufacturers also decide to conduct biomarker of exposure studies. This is to clinically demonstrate that the reduction in HPHCs observed in the aerosol also translates to the clinical setting. Another clinical observation that is often carried out together with the PK and PD studies is called user topography. User topography measures unique characteristics of product use, such as puff volume, puff duration, number of puffs, and flow rate. These are all important parameters that are input into the preclinical risk assessment to get a more accurate and product-specific indication of aerosol dose during inhalation. Behavioral studies are vastly important, and many would argue are the single most important data point in a regulatory submission, especially in the US. FDA are rightly extremely sensitive to the underage use of ENDS products. As I described in an earlier video, significant on-ramping amongst youth can be highly detrimental to the harm reduction potential of your NGP. Behavioral surveys are designed to determine the risk perception and the intent to use your product amongst nationally representative samples of the US population, amongst youth, young adults, adults and older adults. FDA would use such data to determine the risk that your new NGP will encourage on-ramping if introduced into the market. As well as the product itself, other characteristics like color, name, form factors all play important roles in this assessment. Together with the reduced risk potential data, as described in my previous video, the clinical and behavioral data are the areas in which the regulators are likely to focus when assessing an application. Having strong and comprehensive clinical and behavioral data will allow your applications to address all aspects of the modified tobacco harm reduction equation and provide regulators with the confidence that your NGP will positively contribute to population-level tobacco harm reduction.